Okay, so we're going to be talking about transcription, translation, and protein synthesis. So these are just really important processes and pretty much are just like how proteins are made. Um, so proteins, if you remember, are made up of amino acids. Hundreds of amino acids make or link up to make a protein, and there's 20 types of amino acids. So there's infinite combinations of amino acids, and there's, proteins can be of infinite length. There's so many different combinations of proteins that can be made. So protein synthesis is the process by which our cell takes the DNA and converts it into a protein. So there is a problem, and that is that DNA is only found in the nucleus, and proteins are only made outside the nucleus or in the cytoplasm. So the reason this is a problem is because we can't take that DNA outside of the nucleus, and we can't really make the proteins inside the nucleus where the DNA are. So the way we solve that problem is using another messenger called RNA. So it's very similar to DNA, but it has a few differences. If you remember from what we covered when we talked about biochemistry and major biomolecules, you'll remember that RNA is single-stranded. It contains the base uracil instead of thymine, and it's only made of one strand, and its sugar is ribose instead of deoxyribose. And the reason we can use RNA is because it's single-stranded, so it's much easier to take the messages from the DNA and transfer those messages to the ribosomes in the cytoplasm. If you remember from, again, our biochemistry class, you'll remember that ribosomes are protein factories. They make proteins. So the RNA takes the genetic instructions coded in the DNA and transfers them to the ribosome. So there's three types of RNA involved with the making of proteins, and that's mRNA, it's the first one. It's the thing that actually carries the message from the DNA to the cytoplasm. Then you have tRNA, and this transports amino acids to the mRNA to make a protein. So note that these different types of RNA have different structures and different lengths. The final type is rRNA, and it makes up ribosomes, which actually make the protein. So ribosomes themselves, just to clear this up, are made of proteins and RNA. The RNA is like the major part of the ribosome, and the protein is kind of just structural. So ribosomes are really just, spheres of protein that support the RNA. So these are the differences that I just mentioned about RNA versus DNA. It contains a ribose sugar, that's why it's called ribonucleic acid, instead of deoxyribonucleic acid, which is what DNA is. It contains a uracil instead of thymine at the base, and it's single-stranded. And the key word here is usually, because tRNA, this tRNA, actually has a looped domain, which pretty much means that the RNA actually binds with itself. And you'll see that when we actually look at the structure of tRNA. Yeah, this is the structure of RNA. You can see it's single-stranded. You can see it contains uracil, and you can't really see the sugar. So there's two major steps in protein synthesis. The first is transcription. So transcription is when the genetic information from a strand of DNA is copied into a strand of mRNA. Then there's translation. Translation is when the mRNA goes to the ribosome, and that forms a protein. So actually, in between transcription and translation, we actually edit the mRNA to make it more efficient, and we add certain things to it, take away certain things from it. to make it protected against degradation, and also just to make the process of making proteins more efficient. So we'll talk about that. So another term that we have to know is central dogma. So central dogma is just the order of events. So note that it goes from DNA to RNA to protein. This is how information is transferred. Starts in the DNA, then goes to the RNA, then goes to the protein. So this is the central dogma of molecular biology. So transcription. So transcription, if you remember, we talked about DNA replication, and in DNA replication, you had the chromosomes unzip, all the DNA unzip, and it was replicated, and it created 2 new strands. So I don't really want to talk about it in words. I think it's better explained in an image. So this is RNA transcription right here. So RNA transcription is mediated by an enzyme. Maybe I'll open this in a tab, see if it popped out. Transcription is mediated by an enzyme called RNA polymerase. Now, RNA polymerase, if you remember from our discussion on DNA replication, that uses an enzyme called DNA polymerase. RNA transcription uses an RNA polymerase. So the RNA polymerase copies the DNA strand, as you can see it's doing here, and it's creating a new strand. This new strand is called the RNA transcript. So some important things to note about this process. The first is that if you look at the direction of the strand, it's 3' to 5'. The direction of the strand being created is 5' to 3'. This is really, really important that the mRNA transcript that's produced is always in the 5' to 3' direction. That is really, really important. So if you're given like a strand of DNA and you need to find the mRNA transcript for it, make sure that the mRNA transcript you create is in the 5' to 3' direction. If it's not, if it's in the 3' to 5', then you kind of just have to read it backwards. Always start with the 5' end. Also note that there's 2 different names for the strands here. The strand that is being copied is called the antisense strand. The strand that is, kind of just doesn't have anything to do with the transcription is called the sense strand. The reasons, one is called the antisense and the other is called the sense is because if you actually look here, the antisense is the opposite of the RNA transcript, while the sense strand is the same as the RNA transcript, except for the fact that all the thymines are replaced with uracils. But otherwise, the sense strand has the same genetic makeup as the RNA transcript. That's why it's called the sense strand. That's why the antisense strand is the template for the RNA transcript. So going back to here, you see that, DNA is unzipped, then the base pairs pair up, and then, we form a new backbone. So we don't need to watch the animations. So now let's have a question for you. So what RNA strand will be made from the following DNA sequence? Just to note, assume that this is the 5' end, this is the 3' end, just make it simple. So, this is the 3' end and this is the 5' end. So that when you transcribe it, this should be the 5' end. That'll be the 3' end of the RNA transcript. Also, just to make it easier for you guys, don't do the entire sequence, just do what I've highlighted here, TACGCA, because if you can do this, then you can definitely do the entire sequence, and I don't want to make you guys do that much work. So just figure out the RNA strand from this highlighted sequence here. Again, you don't have to do the entire sequence. You can just do the first 6 bases if you want to. Okay, so this is the answer. Note that's literally just a direct copy of the strand shown here. Remember that you just have to know what nucleotide binds with what. So thymine binds with adenine. Adenine will bind with thymine if it's DNA, or uracil, it's RNA, so it binds with U. And cytosine is going to bind with guanine. And again, guanine binds with cytosine. Then you can just continue that throughout the whole sequence. So yeah, okay. So now let's talk about RNA editing. So an mRNA molecule has to be edited in order to actually be used. This is because there's a lot of unnecessary information that needs to be removed. And this unnecessary information is often called junk DNA. We actually have a lot of it, so that's why it needs to be removed. So an mRNA sequence that does not code for protein is called an intron. I don't know why they said interon. There's no E. So if it does not code for the protein, it's called an intron, and a sequence that is useful in making a protein is called an exon. So this is RNA editing. You have your DNA, and the DNA is converted into your RNA. Note that the RNA has exons and introns, but we don't want the introns because the introns are kind of, I guess you could say, junk DNA. They're not actually going to code for our protein. So what do? We remove all of the introns and we create a chain of exons. So you just have a chain of exons and we remove the introns. So that's what RNA editing is. And as I said, this makes RNA processing and turning it into a protein a lot more efficient because it's less RNA that actually needs to be turned into protein. So now let's talk about translation, which is the second step of our mRNA processing into protein. So now that our mRNA molecule has been made, it's time for its message to be made into a protein sequence. So how does the mRNA sequence actually translate into an amino acid? So there's one problem. If you remember, there are 20 different amino acids, and we said there are only 4 different RNA bases. So we have to, if these are all of the amino acids, well then if we just use each RNA base to code for a single amino acid, then it's just, we're only going to be able to code for 4 amino acids. And, we have to code for 20 different amino acids. So, we can't just use one base to code for each amino acid. What we actually do is we say each amino acid is coded for by 3 different nucleotides. So as you can see here, you have, one amino acid being coded for AUG, the second is coded for CGU, and so on and so forth throughout the entire mRNA sequence. So these, little sections of 3 nucleotides are called codons. And codons are what codes for the amino acid. So, this is, a classic diagram that you'll see, and it shows what 3-letter code codes for what amino acid. So if you notice here, you can see that multiple codons can code for a single amino acid. And, this is because if you do some math, you'll see that there are 64 different possible combinations if you're going to use 3 different nucleotides. But we know that we only have 20 amino acids. And what this means is that, we're going to have 44 extra codons because 64 minus 20 is 44. So those extra codons are distributed among all of the amino acids. So some, will have 4, some just have 1, some have 3, some have 2. So, different numbers of codons code for different amino acids. So this is the genetic code. So now let's say you're given a sequence like this, right? AUG, CGU, ACU, GAU, CGU, and so on. Let's say you wanted to actually, make this into the amino acids and figure out which amino acids there are. Well, what you're going to do is you're going to come down to the table here, and you're going to see, okay, first amino acid I see is AUG. So let's take AUG and let's put it into the codon table. We see that AUG is here. So this is the first letter is A, second letter is U, and third letter is G at the bottom here. So it's going to be right here. AUG is met, which is the amino acid methionine. So this would be methionine. Now you can continue doing that. You can take CGU. Now, first letter C, second letter G, third letter U. So it's going to be here, CGU. And that's going to be arginine. So your sequence, as you're building it, is now methionine, arginine. Then you can go on to the next one, ACU. ACU, again, A, first letter C, second letter U, third letter. It's going to be threonine as the next amino acid. So what I'm going to do is I'm just going to copy the next, let's say, let's do 2 codons into the chat. So, and then I'll pull up the chart on the table so that everyone can see it. So here's the chart. So if you guys want, take those 2 codons and figure out what amino acids they will code for. You can just type in the 3-letter word for each. Okay, so the answer is going to be asparagine and then arginine. So, I think that gives you the answer here. Yeah. So asparagine, then arginine, and you can continue with all of the rest of the, the amino acids. So the final thing that we have to notice here is that there's certain, different, I guess you could say, intricacies when doing this. So the first thing is that this table only provides you the genetic code in the 5' direction. What this means is that this first letter is the first letter in terms of like the 5' sequence. So if this sequence that they gave you, if this was the 5' end and this was the 3' end, then you would start from over here. So your sequence would then be AGU, UAG, ACU, and so on for the rest of the amino acid. So your first amino acid would be AGU, which is serine. So if this was the 5' end, this would end up being serine. So that's something important to note, that you have to make sure that your sequence is in the right direction to use this codon table. The other thing is that you see here stop. Like, why does it say stop for certain codons? And the reason for that is because, some amino acid, or sorry, some codon has to tell the ribosome that, it's time to stop. You've done the entire protein sequence. There's no more that you need to do. So the genetic code actually contains these stop codons, and this makes the ribosome stop translating. So what you're going to get is, the mRNA is just going to unhook out of the ribosome, and it's just going to be released into the cytosol, or it'll be released into the endoplasmic reticulum for transport. So step 2, translation, you can see that the UGA right here codes for the stop codon. And then, this could also be UAG or UAA. Now, the other thing is that you'll notice that the only amino acid that has one codon is methionine, or the 3-letter code is just MEP. And the reason for this is because methionine is the start codon. What does this mean? This just means that, like, these sequences being stop codons, this tells the ribosome to start translating. So, this mRNA sequence that we actually end up producing, right? Let's go back. So this this RNA that we actually end up producing, these exons sometimes contain, regions in which, you can't start translating right away as soon as you get the mRNA. You have to look for the first methionine. So once you see the methionine, then you will start translating. So that's the start and stop codons with mRNA. So just as a quick recap, you'll see that DNA is going to be transcripted into mRNA. Then it's going to be turned, edited, turned into mature RNA. Then it's going to be moved into the cytoplasm. It's going to attach onto a ribosome. The ribosome is going to bring in, the tRNA. The tRNA brings the amino acids to the site to build the polypeptide chain. So image this shown here. Okay. So as you can see here, you have your translation going on. This is kind of an enlarged view of translation. You can see the ribosome is moving along the mRNA, and this polypeptide chain, which kind of sticks out of the ribosome, continues getting larger. Then eventually, the completed protein molecule, it just breaks off of the ribosome. Now, this is a more, in-depth view. So what you can actually see here is the first thing is you see the tRNA. So just note that this is the tRNA. So whenever you see this, this is going to be a tRNA. This is the mRNA sequence, and this giant orange thing is the ribosome. So you can see that the ribosome is going to be moving along the mRNA sequence and translating it. So some important things that we did mention here. The first is that the tRNA, it contains something called anticodons. So the anticodons, actually match up with the codons and are complementary to them. So there's 20 different tRNAs, each with a certain anticodon. So let's say you're making like a protein, and the protein, needs an AAG or has an AAG codon. Well then, a tRNA is going to recognize that, bind to it, and become activated. Then it's going to go out and seek the amino acid that's actually the same, or that's going to bind to that specific tRNA, which in this case is lysine. So, the anticodon is very important because it gives an identity to the tRNA and pretty much says, what amino acid are you going to hold? So the final thing is that, the tRNA is reused. So like the tRNA is going to come, from wherever it is in the cytoplasm. It's going to bind to its amino acid. It's going to come over. It's going to, go into the ribosome, and then it's going to the anticodon is going to attach to the codon. Then the tRNA is going to move over, and then it's going to attach its amino acid to the polypeptide chain, making it grow. And then the tRNA, this is where it's going to attach its amino acid. Then finally, it's going to be ejected, and it may be reused. Another thing, with our transcription, that when it goes on, you can actually have multiple ribosomes on a single mRNA strand. So, for example, some cells, like the pancreatic beta cells, which release insulin, insulin's a protein, those pancreatic beta cells, they create a lot of insulin, which is just a protein. So, the DNA and RNA polymerase can only work so hard. So they maybe create, 10 to 100 mRNA transcripts, but each of those mRNA transcripts is reused like thousands of times by, the same ribosomes, different ribosomes. Sometimes you can have two ribosomes on the same mRNA, and this really helps to enhance the amount of protein being produced while also decreasing the amount of the work of the cell. So mRNA can be reused over and over. So today was a shorter class. Sorry about that. We're just going to talk about mRNA, transcription, translation, and all that. So if you guys have any questions, please ask them now about this entire process. Okay, so if there's no questions, then that'll be the end of the class. Thank you guys for attending. I know it was a shorter class. I mean, we got through the information kind of quickly. So nothing we can do about that. But thank you guys for attending, and I will see you guys in our next class.